Hello, my name is Tiana Scott. I am an undergraduate at Brigham Young University, and today I will be presenting clinical exome sequencing data that reveals high diagnostic yields for congenital diaphragmatic hernia plus and new phenotypic expansions involving CDH. So clinical exome sequencing is a powerful diagnostic test that sequences the protein coding regions of all 20,000 genes in the human genome. It is commonly used to identify the causative gene in individuals with multiple congenital anomalies, but it has not been universally adopted as a diagnostic test for individuals with CDH+, which is defined as individuals that have congenital diaphragmatic hernia plus at least one additional congenital anomaly. In order to determine the diagnostic efficacy of exome sequencing in diagnosing individuals with congenital diaphragmatic hernia plus, we analyzed data from 12,000 studies. And so out of those 12,000 studies, we identified 76 individuals with CDH+. Out of those 76 individuals, we were able to find a molecular diagnosis, meaning that it is a definitive or very probable diagnosis in 37% of cases, and a provisional diagnosis, meaning that we're pretty sure it's that gene, but can't say for 100% whether or not that's the cause in 9% of cases. And so this data has led us to conclude that exome sequencing should be ordered for CDH+ cases when a specific diagnosis is not suspected and other genetic testing is negative. And so with that data, we also wanted to, as well as identifying the diagnostic efficacy, we wanted to identify new susceptibility genes that cause CDH. And so traditional CDH gene discovery is done through research and clinical tests. This involves identifying CDH candidate genes and then gathering individuals with CDH caused by variants in that gene in order to determine if that gene is truly causative of the CDH in these individuals. This is a great method for identifying disorders and genes that cause high penetrant CDH, meaning that just about everyone with that mutation has CDH. Clinical exome sequencing data can help us identify human disease genes, especially those that have a low CDH penetrance, meaning that not everyone who has a mutation in that gene has CDH. This can also help us identify phenotypic expansions, which is when we add an additional feature to an already established syndrome. So this means that we identify genes that are already linked to a genetic syndrome, but are also linked to CDH and have low CDH penetrance, which means that we can add CDH as a feature to that syndrome. And so in order to do this, we take all of our clinical exome sequencing data, and from that data, we pull the solved cases that have CDH+. In the data from these solved cases, we'll have 3 different types of genes that we can see, well, mutations in genes. And so we'll see mutations in genes that are known to cause CDH+, and we'll see mutations in genes that don't cause CDH+, and the mutation is just there out of chance, because all of us have some chance mutations that are scattered throughout our genome. And then we'll have our third category, which is the category that we're looking for, which are the new human disease genes that cause CDH+. So the big question is, how do we distinguish between these last 2 groups? How do we distinguish between those genes that are caused just by chance and those genes that represent bona fide new CDH+ genes? And so there are many ways that we can distinguish between these 2 categories, including we can look at whether or not they're expressed in the diaphragm, and because we expect that those genes that cause CDH will be expressed in the developing diaphragm. We can look at published case reports to see whether or not others have identified a link between mutations in this gene and CDH in the past. And we can look at mouse models to see if the mice that have mutations in this gene also have CDH. And the last way that we can distinguish between these 2 groups is through machine learning. So machine learning, we use a computer to try to identify new CDH genes under the assumption that our new CDH genes will be similar to CDH genes that have been previously identified. And we assume that they'll be similar in many different ways. So we assume that they'll be similarly annotated and that they'll be expressed in similar tissues and at similar times during development. So we would, for example, in CDH genes, we would assume that most of them will be expressed in the developing diaphragm. We can also look to see if their protein products interact, and meaning that they're part of the same pathways and therefore cause the same or similar syndromes when they're mutated. They're targeted by similar microRNAs or regulated by similar transcription factors, so they're turned on and off around the same time, or they have similar epigenetic modification patterns. And then we can also look to see whether they cause similar defects in mice. And that's a lot of different areas of this gene to look at to see whether or not they're similar. And so in order to get all of this information, we use many knowledge sources, so many public databases where all of this information has been stored, and we can access these and then look to see if we can find patterns that are present between all the different CDH genes. And so when setting up our machine learning algorithm, we have to get a training set of known CDH genes in order to train the algorithm. And then we can validate through a leave one out cross validation and a second validation in a set of CDH candidate genes. So our training set was pulled from research that was previously done and consists of all of these genes. And so once we had trained the machine learning algorithm with this training set, we then moved on to our leave one out cross validation. So this is a validation that works by removing one gene from the training set. So for instance, we remove chat from our training set of genes, and then we run the algorithm with this training set, after which we run all of the ref-seq genes and chat through the machine learning algorithm in order to generate a score. And so this generates a rank score, which compared to all ref-seq genes for that training set gene. So for instance, chat has a rank score of 91.4%, meaning that it was more similar to the training set than 91.4% of all ref-seq genes. We can then return that gene to our training set. So we put chat back in and we remove the next gene, run the algorithm, and then generate a score for that next gene compared to all ref-seq genes. So in this case, DNA SE2 has a rank score of 92%, meaning that it scores higher than 92% of all ref-seq genes. And then we can return him and pull out the next one and keep going to generate scores for all of our training set genes. And so when we look at these scores on a graph, so here we have the fraction of training genes with higher scores versus what percentile of ref-seq genes they scored higher than. And so if the machine learning algorithm was assigning scores randomly, we would expect a distribution along this dotted line. But as we can see, instead, the distribution is along this curve above the dotted line. And the area between that curve and the dotted line tells us how well our machine learning algorithm is performing in identifying real CDH genes. And so we have a significant amount of area under the curve, so we see that it is identifying these CDH genes at a frequency higher than that of chance. And so we've looked at our training set, we've done our leave one out cross validation, and move on to our second validation, which uses another set of CDH candidate genes. And so in the second validation, we used 35 CDH candidate genes that were not in the training set that had previously been identified in prior research. And we ran those through a machine learning algorithm to generate scores. And we expected to see they should be scoring higher because they are CDH genes as well. And so when we looked at the results for this test, we found that the median CDH score was 95.5%, meaning the median score for these CDH genes, they scored higher than 95.5% of all ref-seq genes in similarity to our training set. And all but one of these genes had a score higher than 50%. So this shows that our machine learning algorithm can identify bona fide CDH genes even when they were not originally part of our training set. And so, like we said, there's many different ways that we can distinguish between those mutations that are by chance and phenotype. So we used the machine learning algorithm, and we have generated scores for all of the genes that we found during the study. And then we also looked at the other ways to distinguish in order to further verify whether or not these could be CDH genes. So we looked at the expression in developing diaphragm, published case reports, and mouse models, as well as our machine learning algorithm scores. And we identified several genes that appear to be new CDH genes. And so some of these genes include FOXP1, which is associated with mental retardation, with language impairment, and with or without autistic features. FOXP1 had a machine learning score of 96.9%, and it is also expressed in the developing diaphragm. We also found other cases in the literature that found that where FOXP1 was associated with congenital diaphragmatic hernias, which all support our conclusion that FOXP1 is a new CDH susceptibility gene, even though it was not seen, CDH was not seen in mouse models for this gene. Another gene that we found is EP300, which is associated with Rubinstein-Taybi syndrome 2. This gene had a machine learning algorithm score of 94.7%, and 2 individuals from our cohort of 76 had mutations in this gene. Also, this gene is found to be expressed in the developing diaphragm, and we were able to find other cases in the literature to support this connection between EP300 and congenital diaphragmatic hernia, which leads us to conclude that it also is most likely a CDH susceptibility gene, even though CDH is not seen in the mouse model for this gene. SMARCA4, which is associated with Gofin syndrome 4, had a machine learning algorithm score of 93.7%, and it was seen to be expressed in the developing diaphragm, and other cases in the literature have also shown a connection between SMARCA4 and congenital diaphragmatic hernia, which once again leads us to conclude that this gene could be a CDH susceptibility gene, even though we do not see CDH in the mouse models. SMARC-C2 similarly causes Gofin syndrome 8. It had a machine learning score of 89.9%. It is expressed in developing diaphragms, and there are other cases in the literature that support this connection, which again leads us to conclude that it is a CDH susceptibility gene, even though we do not see the CDH in the mouse model. And the last gene that we wanted to talk about is ALG12, which is associated with ALG12 congenital disorder of glycosylation. This gene there were 2 individuals in our study that had mutations in this gene and congenital diaphragmatic hernia, and it is seen to be expressed in the developing diaphragm, but it only received a machine learning score of 48.2%, which is a lot lower than the other genes that I've talked about today. So even with this low score based on other lines of reasoning, we conclude that it is a CDH susceptibility gene, but the score might be lower because it might be affecting a slightly different pathway than the other CDH genes, and therefore isn't quite as similar as we would expect. Or it could be that the data that is in our machine learning algorithm is not quite up to date enough to catch the similarities that are found here. And we're actually working on an update for our machine learning algorithm that hopefully will be able to better identify genes such as ALG12 that are scoring a little bit lower than we would expect, but are still seen to be associated with CDH in some way. And so as of now, we've identified 13 potential CDH susceptibility genes based on this machine learning algorithm. We talked about 5 today, which are highlighted in green, but there are others on this list that we also found that have similar supporting evidence as to why they could be CDH susceptibility genes. And so we would like to thank all of those that have helped in this research and made it possible, especially Baylor College of Medicine, Brigham Young University, Texas Children's Hospital, and the National Institute of Child Health and Human Development. Thank you all for your support, and thank you so much for listening.
Hello, my name is Yoel Gofin. I'm a medical genetics fellow at Baylor College of Medicine. Today, I will present some evidence that indicate the role of the gene FGFRL1 in congenital diaphragmatic hernia. When we talk about chromosomal abnormalities, there are two main types, deletions and duplications. A duplication is an event where a segment of DNA appears twice, and a deletion, on the other hand, is when a segment is missing from the chromosome. There are other types of abnormalities which will not be discussed in this talk. There are some genetic syndromes that are caused by specific deletions and are associated with congenital diaphragmatic hernia, or CDH. There are Pallister-Killian syndrome, 1q41q42 deletion syndrome, Wolf-Hirschhorn syndrome, and others. CDH is also associated with chromosomal aneuploidies, where a whole chromosome is missing or appearing twice, like in trisomy 13, trisomy 18, and trisomy 21, known as Down syndrome. So what test do we use to diagnose chromosomal abnormalities? The method used today is called chromosomal microarray. In this test, we use sample from a patient, colored green, and another sample from a healthy control, colored red. In a normal state, we will see the yellow color, meaning there is equal amount of DNA on both samples. If a certain segment is missing in the patient, we would only see the red color. If, on the other hand, a patient will have a duplication, the green color would be stronger. One of the known chromosomal abnormalities associated with CDH, as mentioned, is Wolf-Hirschhorn syndrome. It is characterized by intrauterine growth retardation during pregnancy, and after birth, we see failure to thrive. There are typical craniofacial features that resemble a Greek warrior helmet with a broad nasal bridge. There are seizures and cognitive impairment, and we see brain anomalies on MRI. Cardiac anomalies also appear, with atrial septal defect and ventricular septal defect as well. There are eye findings and also genital anomalies. This syndrome is caused by a deletion in the fourth chromosome, specifically in the region called 16.3 on the chromosome's short arm. As the region called 4p16.3 is quite large, it contains many genes. What we asked was, from all of these genes, which one is the one responsible for the CDH feature in Wolf-Hirschhorn syndrome. With data from the following two patients, we would like to point out to a smaller, critical, and more specific region within 4p16.3 in the context of CDH. Patient 1 was a female fetus found to have CDH on prenatal ultrasound. There were no other organ malformations on the ultrasound, and the brain imaging was normal. The parents chose to terminate the pregnancy, and genetic testing was taken from the fetus. A chromosomal microarray, which is the technique I mentioned earlier, revealed a de novo deletion, meaning a deletion that did not appear in one of the parents. The deletion was in the size of 1.8 million base pairs, or letters, in the 4p16.3 region previously mentioned. When compared to previous deletions reported in this region in the past, this segment is the smallest reported, and so can point out to the more specific location of the element in Wolf-Hirschhorn syndrome that causes CDH. The next question would be, what is the exact gene that is the culprit in causing CDH in patients with this syndrome? This is a caption of the genes contained in the region deleted in patient 1. As you can see, there are quite a few, and so at this point we need a tool that can prioritize these genes. It is to this point that machine learning can assist, as was presented in the previous presentation. The algorithm first receives genes that are already known as causing CDH, and with that information, the algorithm looks for similar genes and gives them a score as a measure for this similarity. In the mentioned critical region, 7 genes were proposed by the algorithm, rated by grade. The second candidate was FGFRL1. And why do we refer to him specifically? Well, that's because FGFRL1 is the only one among the candidate genes that is actually known to be associated with diaphragmatic anomalies, but up to that time, it was only seen in mice. In a study that looked at FGFRL1 knockout mice, meaning mice without that gene in their DNA, these mice had abnormal diaphragm, and their lungs were not able to fill with air, as can be seen in caption B, that shows a lung that did not float, like the one from the control mouse on A. On the right, you can see the diaphragm of FGFR1 deficient mice. It was thinner with less muscle tissue. Under the microscope, the diaphragm was thinner and hypotrophic, meaning with less growth. Like most genes, we have 2 copies of FGFR1. The question is one copy of this gene enough for a normal diaphragm? For mice, it seems so, since mice with only one FGFR1 copy had normal diaphragm. In humans, a database called NOMAD includes healthy adults. This database showed that there are healthy individuals that have a detrimental change in one gene copy, but with no apparent effect on their diaphragm. This presumption is important when we try to interpret the data from patient number 2. Patient 2 was diagnosed with CDH during pregnancy by ultrasound and MRI. He had a left hernia with only a small residual left lung. He was born at 39 weeks and had normal weight, length, and head circumference with no specific facial features. He was intubated after birth and later had a head ultrasound and a heart ultrasound and echocardiogram, which were both normal. Due to respiratory failure while being intubated, he was connected to an ECMO machine that supplies his blood with oxygen, taking the place of his small lungs. While on ECMO, he had a CDH repair surgery. The description given by the surgeons was that about 95% of the left diaphragm was missing, meaning this was a severe hernia. After a few days, the patient was taken successfully off ECMO, but sadly had an acute decompensation and passed away. MRI and CT performed after his death showed no other abnormalities in his body, which defined this case as a patient with an isolated diaphragmatic hernia. This patient had extensive genetic workup. An exam to find the major trisomies and sex chromosome abnormalities was normal, as was the chromosomal microarray. The additional test he had was called a whole exome sequencing. This is a very comprehensive DNA sequencing method that practically reads all the genes that are currently known, which is more than 20,000, and it looks for spelling mistakes. Of these genes, only a few thousands are currently known to be associated with human disease. The patient's report had no variants or changes in genes that were known to be associated with human disease, but it did describe 2 changes in FGFR1. As findings in this gene were only described in mice up to that time, these variants were not considered in the report as a positive result, explaining the CDH. When we look at the location of FGFR1, it is relatively in the middle of the critical region defined by patient 1. What we can say about the 2 changes found in FGFR1 in patient 2 is that they were seen in healthy adults of Latino origin, like the patient, but never on both copies at once. Of all changes mentioned in the exome report, these were the best candidates to explain the genetic origin for the hernia. While patient 1 had only 1 copy of FGFR1 missing, patient 2 had changes on both copies of the gene. However, generally speaking, not all changes cause damage, and the computer models predicting the effect of these changes were inconclusive. When looking at all this data, we believe that FGFR1 can cause a susceptibility for CDH. So to sum up the information presented here today, the deletion in patient 1's chromosomal microarray defines a critical region within 4p16.3 related to the CDH feature of Wolf-Hirschhorn syndrome. Patient 2 is the first human patient with CDH that was reported to have a change on each copy of the FGFR1 gene, bringing the data known only in mice up to that point 1 step forward towards the role of FGFR1 in human disease. And so, as a take-home message, our study shows that FGFR1 deficiency contributes to the development of congenital diaphragmatic hernia in humans. I would like to first thank the patients' families and also thank Dr. Daryl Scott for his guidance, our colleagues in Torino, and thank you all for listening.